Hello again everyone, welcome back to ptet.chemistry channel. So in this follow-up tutorial video to the other methods of purification or separation techniques on top of what I've covered in the previous lectorials, which was going through filtration, evaporation, crystallization, as well as distillation in details, including the experimental setup. So all those labelings, all those what is wrong with the experimental setup, as well as in fact drawing or sketching the particular experimental setup is very important. Okay, so for example in these questions, so this particular lectorial will go through a couple of questions that derive from the particular concepts I covered in the previous lectorial. Okay, so in this question we have a student who investigates a sample of rock salt. So the student grinds the rock salt into a powder. Okay, so grinds mean break it up into smaller pieces, and you grind it into very small pieces, powders, and you places the powder in a beaker and you add water to it, and then you stir the mixture. Okay, so we apply some force and we stir the mixture to try and mix it, and then we pour the mixture through the apparatus shown. So this is very clearly a filtration apparatus setup. So if you can identify the apparatus, then you can identify the technique. Similarly, if you know the technique, you should be able to draw this from scratch. This is a scientific diagram to represent a filter funnel, and everything will flow through the filter funnel unless you have a filter paper. So this thing there is actually a filter paper supported on top of a filter funnel. Okay, if you don't have the filter paper, all of these can go through because these have holes in between. The filter paper will trap the solid and only allows the solution or the liquid to flow through. So here you see you get a colorless solution. This is called the filtrate. Okay, these are all technical spelling. And the brown solid that is left over on the filter paper after all the solution has passed through the filter paper, you get all the filtrate, then you're left with residue which are left over on the filter paper. This thing is a conical flask, and you use a conical flask because it has a flat bottom and it has this top that can support the filter paper. Alright, so name apparatus A. Apparatus A is filter funnel. So I have just gone through all the apparatus setup instead of just going through for the question. Of course, in a real exam, you probably have one look at the question and you probably just jump straight to the question. But I'm not just doing the question for the sake of it. We're actually interested in learning about the whole apparatus setup because it is actually a whole part of a story, okay? Rather than just talking about a little bit here and there. As I mentioned, a brown solid remains in apparatus A. That is what you call a residue. The residue is what remains on the filter paper, and you get a colorless solution collected. In that colorless solution is called a filtrate. Name the process to separate the solid from the colorless solution. You need to be careful here. The solid from the colorless solution. So we have a solid in here. Okay, we want to separate solid from colorless solution. We don't want solid from the brown solution. So you need to read the question carefully. You get this colorless solution, there is some solid in there. I want to get the solid from the colorless solution. Well, what we could do really is we could evaporate off all the solvent, or we can try crystallization. Alright, so I would imagine that evaporation, or you can try crystallization. So both of these will work because it's used to obtain a solid from a solution. A solid from a solution, a solid substance that is dissolved in the solution from the solution, either evaporation or crystallization. And in your specific learning objective, what have we got here? Well, they don't specify which one, but they like the term crystallization. So you could essentially use crystallization or evaporation. But since the syllabus specified crystallization, perhaps we should use crystallization instead of evaporation. Evaporation means to dryness. Crystallization will be a slower, but it will give you a purer solid in the end. The colorless solutions contain two different cations. Okay, I know what. This one we can skip because you will deal more with this when you cover identification of cations and anions as part of analytical chemistry. So there will be more experimental techniques as part of analytical techniques or analytical chemistry, which we'll talk about another time. In this next question, so I hope you can see these are longer structured questions, and these are probably easy marks to score because it's 2 marks and they give you 3 lines. So they need to be 2 important points of interest that are relevant to what they're asking. All right. So they want to separate ethanol from a mixture of ethanol and water. I talked about this in the previous lectorial, specifically in the context of COVID. Ethanol is an important solvent used in making hand sanitizers. All right, so the world was short of ethanol for some time because people couldn't get to work, and suddenly there's a huge demand for ethanol. So it's very important this particular process to separate ethanol from water because you want the pure ethanol to give you sanitizers that are clean, right? Okay, so this ethanol is soluble in water, as you would know. Ethanol is dissolved together with a little bit of other solvents, but ethanol can also be dissolved in water. That's why sometimes we see 30% ethanol, 60% ethanol. That means the rest are water and it's not 100% ethanol. So we have a mixture. These two have different boiling points. And without going into too much details, you're not supposed to remember the number. Apart from water, the boiling point of water is 100 degrees Celsius. Ethanol has a lower boiling point than water. That is a very important concept to note. Ethanol has lower boiling point than water. And that means when I do distillation, ethanol, which has the lower boiling point, will turn into gas before water. So ethanol will turn into gas before water, and ethanol will be collected first because ethanol will vaporize and it get condensed first. I will collect ethanol and the temperature will be constant because I'm collecting ethanol at its boiling point, which is equal to its condensation point. So it's boiling here as ethanol, so the temperature at which you collect the ethanol will be the boiling point of the ethanol, and it's a pure substance. A pure substance has a fixed boiling point. All right, and then after all the ethanol has been collected, the temperature will continue rising and rising and rising until you reach 100 degrees Celsius. Then the water will start coming out and you collect it in a different conical flask. This one says pure water here because this is not ethanol and water. This is a salt solution, salt and water. Salt has higher boiling point than water, therefore water is collected first. It's different when you have ethanol in water. Different scenario, you have to think about the different boiling point. So 2 concepts there, 2 points there. The first process is called distillation. In fact, we're talking about simple distillation, not fractional distillation. So it's just called distillation. Suitable because, so we just jump straight in. Do not repeat question, okay, because there will be a waste of time. Suitable because ethanol and water have different boiling point. So because they have different boiling points, you can separate out the one with the lower boiling point first. Different boiling points. So we can vaporize. Vaporize means boil. Vaporize means you turn something into a gas from the liquid. Vaporize or boil the ethanol first, which condenses first, because after you boil it, it will condenses first after you pass it through the condenser. Therefore, collect ethanol first. But the key thing really there is the different boiling point. Alright, so next question, what do we have? We need to describe, so with a longer wordy type of thing. So this is 3 marks now, and they give you 4 lines. This is also 3 marks and 4 lines. In each case, a suitable method to separate the named substance from the mixture. Okay, so here we have sodium chloride, which is actually salt, your table salt, the one we use for cooking, and sand. So we know sodium chloride is soluble in water. So this is what I mentioned in my other lectorial before this, and this is insoluble in water. Soluble in water means it's dissolved in water. Insoluble in water means it does not dissolve in water. Here you want pure and you want dry. Okay. So here we just need it to be a pure dry samples. In other cases, you might have seen pure crystals of sodium chloride. Pure crystals of sodium chloride would mean crystallization. Pure dry sodium chloride could also mean evaporation to dryness, or it could also mean you could crystallize it and then you dry the crystal, which means you have to filter a second time. After you filter off the first time, you get rid of the solvent which contains the sodium chloride, and then you crystallize out the sodium chloride, and then you filter the crystals again, and then you dry it again, which seems tedious, especially when the question is just asking for pure dry sodium chloride. So what I'm going to do is I will dissolve the mixture, because the mixture contains both of them. So dissolve the mixture of the sodium chloride and the sand. I will use distilled water. So the reason you're using distilled water instead of tap water, and we are specifying to be distilled, is because distilled water does not contain any other ions, and your drinking tap water might contain other dissolved mineral ions. So that's why in chemistry we always use distilled water, okay, instead of using the tap water, because distilled water has no dissolved ions. No dissolved ions, meaning it has got no other substances in it. It's as pure as just H2O water, okay, unlike your tap water. So dissolve that in distilled water, then filter off the insoluble sand, but we will collect the filtrate. The filtrate is the stuff, is the liquid or solution that pass through the filter paper. The filtrate contains the salt containing the sodium chloride. So I'm going to pursue evaporation here. So we will evaporate by heating the filtrate, meaning the solution, and we want to evaporate by heating the filtrate to remove the water, meaning the distilled water, leaving pure dry sodium chloride, which I can use the formula, but since we haven't really covered formula, by the time we do this particular topic, we can just write pure dry sodium chloride if I evaporate it to dryness. Okay. Next bit is on food colorings. So just to separate the food coloring from a mixture of 3 food colorings, this should remind you of chromatography. So we use paper chromatography. Just make sure you can spell it properly. So it's always a big problem among students who, because they cannot spell the technique properly, they're losing out on precious marks. Okay, and it says describe a suitable method. Okay, so it's describe and you've got to explain. All right, so we use paper chromatography to separate the food coloring, the different food colorings, due to their different solubility. So they are all soluble in the solvent of choice. Here I am not sure if we use distilled water or not, because I really don't know what solvent they are soluble in. So that's why it says solvent of choice. They need to be soluble in it in order to move up the filter paper. Okay, so the more soluble the solute, meaning the substance or the food coloring, the further distance the spot, meaning the solute, will move up the filter paper. Okay, and how do we How do we know they are separated? So there are 3 food colorings there. So we expect if separated, we will expect 3 different colored spots on the filter paper, and this filter paper is specifically called a chromatogram because this is what you obtain from the paper chromatography technique. All right, so you can write the story somewhere along this line, but talking about why it works or how it works, and the name of the technique itself, and what are the results that you're expecting in order to separate out one of the food coloring from the 3 different food colorings, giving you 3 different spots on the chromatogram. All right, just very quickly, question 4 is not relevant here. We are looking at question 5. This is a multiple choice question. We want to separate a mixture of ethanol and water. So this is a very common type of question, ethanol and water. So they mix well. All right, so we are looking at distillation because they have different boiling point. So we know the boiling point of water is 100 degrees Celsius, and the boiling point of ethanol, even though you don't need to know it, is around 78 degrees Celsius if I'm not mistaken, but it's definitely lower than the boiling point of water. If we're looking at distillation, we are looking at an apparatus such as this, where this is a heating mantle. It heats it, it passes through that fractionating column, but the vapor will reach there and then it will condense back down, where the water will go in from the bottom and the water will go out from the top, and the thermometer bulb will be where the vapor enters the side joint, because the thermometer will read the boiling point of the ethanol as the ethanol condenses outside and get collected here. So this is the technique that you want, and not this one. Okay, this is a filtration apparatus, and this one is a gas collection apparatus. Okay, for those of you who don't know, this is what you call a reflux apparatus. So a reflux apparatus has also a condenser. It has water in and water out there, but the problem is there is nowhere for you to collect the condensed stuff, because when this get vaporized, it vaporizes and turns into the vapor up, and then the condenser will cool it back down, so it does not escape, it comes back down, goes out as a vapor, comes back down, condense as a liquid. This has a sideway joint. Sideway joint means you can escape. This is just going up with no sideway joint. You can't escape. The liquid comes back down again. This is a reflux apparatus. You don't necessarily do it for your combined science, but I think it is actually in the O Levels chemistry syllabus as well. Okay. Going straight into the next bit. This is not relevant to this separation technique. So next question is something for what we call distillation setup. So we call this fractional distillation simply because it has a fractionating column. Okay, otherwise you could just call it a simple distillation, but the concept is the same. You heat a liquid mixture. Okay, the lower boiling point substance will boil first and turn into a vapor, and then it will condense by the water. And something is wrong here. You can see the water in from the top is already wrong. It should be water in from the bottom and water out from the top, and this is exactly the kind of question they like to ask you. What is wrong with the setup? Okay, well, the water is exactly what we saw earlier already. Okay, and the other common mistakes that students make is that this must be left open. Okay, so you have a liquid that condenses out. Okay, you do not want to seal this. Okay, if this is sealed, you can get a pressure build up in here, because some of it might not condense in time. Then you will get a gas build up in here, and that is dangerous. Gas, pressure, glass, very dangerous to you. Okay, that's why we use the most basic protection like lab coats, as well as using safety goggles in the lab to protect our eyes and to protect our body as well. Okay, but you really want to minimize pressure build up wherever you can. But it's just asking for what error is the student making. So there's just one error here based on the singular use, based on the singular spelling of the error that they are saying specifically in the question. That's to do with the water being in at the bottom and water out from the top. This is an error number 1 that a lot of students will make. Error number 2, error number 3 is usually here. Okay, so here is fine because the bulb of the thermometer must be where the vapor joins the side. And that's it really. So I'll cover in a separate video specifically on the checkpoint questions, on separation techniques or purification techniques, and they'll be in a separate video. Okay, so thank you for watching, and yeah, I'll see you in the next video. Don't forget to subscribe and share the channel with other people if you think the channel can benefit them, and follow me at ptet.chemistry on IG, FB, FB for Facebook, and Twitter if you use those social media platforms. Alright, see you in the next video.